All right, everybody, and welcome to this first edition of DNA Learning Center Live. We are trying something new here. We recognize that lots of folks are at home, and so what we prepared for today is a sort of test run into some bioinformatics, which is one of the ones that are easy to do from the comfort of one's own desk. So I'm going to take you through it, give you a little bit of sort of our intention on what DNALC Live will be, and then actually give a full lesson. If you are watching this on the DNA Learning Center homepage, so we have DNALC, the homepage /resources /DNALC Live, there will actually be some downloads, and I'll direct your attention to that a little bit later. Make sure you know where that's at. And then also you want to check that place again in a few days as we actually populate that. But to get started, let me actually share some slides because I do have a presentation that will go to some of that background, and then we'll go into the actual lesson. Because you probably haven't seen the background or the DNA Learning Center site, this is a bioinformatics lesson, which is targeted at high school and above. So if you're enrolled in, or if you completed AP Biology, you're probably already set. It's also interesting, I think, for undergraduates. We'll be working with some computer tools, and we will have actually some downloads and packets. So let me share my screen because I have that all written down for us, and we'll go from there. And of course, this will be recorded so that as we go a little bit further, if you need to watch this, since this is part one, you can always go back. Okay. All right, so as I said, this is an experiment. We're really happy to try this out, and we're going to be looking for your feedback, your comments. We'll tell you a little bit more about how to get that feedback to us, but certainly through our homepage and through the comments on the YouTube live video. The goal for this whole channel, which we think it will be, is again to provide the same standards of instruction in genetics, molecular biology, bioinformatics learning that the Learning Center has always done. We're going to have laboratory and computer demos, short online courses for middle school, high school, and the general public. We want to also have interviews with scientists, help for teachers, especially transitioning to a lot of virtual teaching and looking for content and resources. And we also have some at-home activities, things that you can do with stuff that you may have lying around your kitchen. Perhaps at one point we'll also have kits, some social media content. So all these types of things we have in mind, and we'd love to hear back from you a little bit more. So really important. Perhaps you're already watching this at dnalc.cshl.edu/dnalc-live. Bookmark that site because that's going to be a great place to go back and see what we have planned next. Also, be sure to like, subscribe, and share on any of our various social media platforms. You're going to see a lot more activity there as well as we post content and as we also communicate with you. I also want to recognize that probably a lot of people that are watching are not just our normal community of visitors to the actual physical learning center here in New York, but actually you may be coming from all over the world. And in fact, the DNA Learning Center is already over the world, so that's great. There are a number of groups that we've collaborated. And if you're not familiar with Cold Spring Harbor Laboratory, of which we are part, it's also just across the street here. We are in New York on the North Shore of Long Island, and it's considered one of the world's top institutions in molecular biology and genetics. So just a little background. I hope you can learn a little bit more on that. So let's get into the lesson. There are a number of parts that we have, a number of goals for today. I'm also going to be getting some assistance from folks who are helping me with questions. So if you're watching us, you may want to make sure that you're actually watching us from YouTube Live. Then you'll be able to take care of the chat function or take advantage of the chat function. All right, who is this course for? So this is actually designed as a course. As I said, we're going to have a variety of YouTube formats and video formats. Some might be really short and fun, but we also recognize that people want to do some substantial learning. And so this is actually designed as a 1-hour course. So who's this course for? And should you, want to get the full benefit from it? So I've targeted this particular course at US AP Biology students. That's usually grades 10 and above. And it's also useful, I think, for intro undergraduate biology. So if you're first or second year taking bio, this is probably going to be something where you have enough background that you're in the right starting place. Of course, anyone can watch the videos, and hopefully everybody gets something out of it. This particular course is going to be done in 3 sessions, 1 per week. So I hope you'll come back to us next week for the next section. This is part 1, and I anticipate that they're going to be somewhere around 45 to 60 minutes each. There are also going to be exercises, so we don't want this to be just a course that you are only listening to me. You're welcome to do that. In fact, you may want to watch and listen to this whole thing first, and then go to the packet that we prepared for you. I'll show you where that is to make sure you can download it. You can also follow along using the online bioinformatics tool, which I'll introduce later, DNA Subway. So you'll have all those resources so you really can actually feel like you're accomplishing something. I'll also mention that we have worksheets for students, and we also have worksheets for instructors, for teachers. So if you want to use this as an assignment, we'll also make those available. We've actually talking about something called DNA Barcoding. So we're actually going to be talking about how can you use DNA to identify an unknown organism. How do we actually understand, once we have DNA sequence, well, how do we actually determine, is that a good DNA sequence? Can we use it to actually identify an organism? How does that even happen with sequencing? We'll talk about that. We're also going to use BLAST. That's going to be in part 2, so not today, to talk about how do we compare unknown DNA sequences to known sequences. And that actually satisfies one of the AP Bio lab requirements. So you may really be interested in that if you're trying to do AP Bio. And we're also in the later section, probably more in part 3, going to talk about comparing DNA sequences using phylogenetics. So we're going to be using this tool called DNA Subway. It is a tool that you can access for free. We do suggest that you get a free account at cyverse.org. You even have time to do that while you're watching me, but even if not, you can come back later with the packet and you'll have access to all of that. Actually, to follow along, you don't even need an account. You can use the guest feature, and I'll be showing that as we get a little bit further along. Okay, so here is the first subject that we need to talk about. Barcoding bioinformatics, this part 1. We're going to talk a little bit about a background, both what is bioinformatics, this term that I've used several times now, and what is DNA sequencing, because we need to understand how do we get DNA sequence, what is that, what's the relationship with DNA barcoding and bioinformatics, and that'll lead us into the actual exercises that we have planned, and that we'll have some questions for you to answer as we get further along. The steps that we're going to do, we're going to introduce bioinformatics, get the background, learn about our example experiment. We're going to be trying to identify some mosquitoes, and we're going to start that experiment and at least get to the quality section. So what is bioinformatics? Well, I've taken this from one of my favorite places, which is Wikipedia. They give you a book version of bioinformatics, which is fine, and you might as well have it if I'm going to ask you the question. Bioinformatics is an interdisciplinary field. And it really talks about storing, retrieving, organizing, analyzing biological data. So that's actually the keyword, biological data. And although many people in the classroom may have used microscopes, if you come to the Learning Center, certainly use pipettes, but the computer is actually one of our most important tools in biology. Sometimes when I'm introducing it, I talk about computers. This is not a CS course, but this little video, which I've taken, it's credited from YouTube, is actually a computer. It's called the Turing machine. Probably the only thing that you might get out of this little video is the sequence of zeros and ones, which this computer is able to draw. Now, this is not an ordinary computer, although it actually can do every single thing that any computer you've ever used in your life has done. It draws zeros and ones, and that's roughly equivalent to what happens inside of a real computer. The problem is, because this computer is pretty big, it's macroscopic, is that if you ask this computer to calculate something or maybe display an image or a video, it would literally take months or years to be able to give that to you. So it's very, very slow compared to how if we're just operating on the level of electrons at the size of our normal microprocessors, it's very slow. But the reason why I also show this video is because I think there's a really deep connection. Here's another video from the Learning Center. It's a deep connection with biology. If you look at this 3D animation of DNA being copied, it also is a sequence of, well, not zeros and ones, but of A's, C's, T's, and G's. And that analogy is not just a surface analogy. It really has some deep relations. There's a whole one-week summer camp called Biocoding, which I hope we run this summer. That goes more into that, but bioinformatics and biology really, really have this deep connection. It's those not zeros and ones, but A's, C's, T's, and G's. So I'm assuming that you have a little bit of background about biology. Here, of course, is that familiar double helix that we're used to thinking about. But that double helix, that sequence of DNA is information, it's data. And it's those A's, C's, T's, and G's that make you up. I think you know the basics if you're watching this and you've taken your AP Bio. How do we actually get those A's, C's, T's, and G's? How do we actually get that DNA so that we could carry out some experiments at the level of bioinformatics, at the level of working with data? Well, that's what's coming up. So when we are speaking about data in biology, we're often talking about DNA sequence, but there are lots of other data. We're just not going to talk about that today. So there's a whole bunch of other things that is bioinformatics that's not necessarily DNA sequence. We're not talking about that right now, but just so you know. Okay, so actually right away, I do want to take you to see some of this at a very, very sophisticated level. There is a resource online called the National Center for Biotechnology Information, NCBI. We're going to have a lot more to do with this in part 2, but I just wanted to show you this element of biology that you can actually just Google and use. So what I'm going to do right now is I'm just going to go ahead and click on or copy one of these links. I thought that they would copy for me, but I'll go ahead and start a new browser. So let's go ahead and open up an entire new clean window there and go to NCBI. Now, doesn't look all that fancy right now, but the cool thing I want to do, just to give you a little bit of an example of this, is if I go here and go to genome, and of course, if you go to NCBI or Google for it, you could also follow along. If I click on the human genome, I'm going to do something that's really, I think, personally pretty fantastic. A number of years ago, depending on where you were born, maybe before you were born, maybe just if you're in sort of the 10th or 11th grade around the time that you were being born, there was a sequencing of the human genome. And what I really think is fantastic is that I can come to this website, and if you know about the human genome, we've got 23 pairs of chromosomes and also the sex chromosomes, mitochondrial genome, but I could click on any one of them. And when I click on it, give it a second to load, what I'm actually going to be able to do, let's go this way. Whoops, I went backwards instead of forwards. What I thought I was going to be able to do, there we go. It's just a little bit slow today, and actually maybe a lot of people are using NCBI. I'll tell you why in a moment. I get all this, which is a mess. It's really, really hard to look at. But if I actually tell you just a little bit about what you're seeing, this is essentially a parts list. This is a map of the human genome. And usually when they load this, they put me on a gene that's interesting. So let's see if they've done one this time. This is a pseudogene. What is this gene? Sometimes they pick really interesting ones. Ribosomal protein. This is all a blur. Take the next course that we're going to do on genome annotation. But if I click on this little bit of information, what it's going to take me to, once you learn how to read that, is actually the DNA sequence of any given unit of DNA that I'm interested in. In fact, going back to my slides here for a second, one of the things that it was trying to center on P10, which is an interesting gene. Actually, I used to work on that, which is involved in cancer. What I wanted to point out today, especially many people are talking about the coronavirus. That's why everybody's sheltering in place. If we actually look, what makes this super, super cool to have bioinformatics is that information about even something that has just come out and just being described and just being found, like the coronavirus, can be shared with scientists all over the world. And it's because of bioinformatics that we're able to organize that information and look at it. So when I come to this page, is one of the first papers that published the sequence of this. If I click on this link here, it actually takes me back to NCBI, but it actually takes me to the whole sequence. These are the proteins. If I go back and click on FASTA, it takes me to that sequence, that nucleotide sequence of this coronavirus. And if you look, I'm not scrolling all that long. I'm at the bottom of it. It's just this one page of information that a lot of scientists are looking at right now. And before the human genome was done, before DNA sequencing was a thing, and we're going to talk about sequencing next, this would have been impossible to do. So while we're in a nasty situation, so to speak, for the moment, it's really, really a time in biology of which we can react and we can know what we're looking at and have an attempt to be able to do something about it. That's what bioinformatics does. So what are we going to do, and what piece of bioinformatics are we going to explore today? Perhaps we'll also develop a nice new lab on looking at the bioinformatics of coronavirus and other viruses. But today we're going to bring it to something that is a little bit more general and that will give you some of the basic principles that you need. It's also connected to some other really exciting experiments that maybe some of you have done, or if not, that I hope you know about. So let me go here. How can you identify an organism using just its DNA? This is a super cool question. How can you actually know that if you have an organism where you don't know what it is, how can you actually get the DNA out and figure out what it is? Well, let's take a look. What is DNA barcoding? And I've got this wonderful image of a passion flower there. Now, give me a second. I just want to actually change something on my computer off-site here, because I also want to be able to make sure that I'm seeing any questions that come across, and we'll be helping with those. So let me give you the steps first, and then we'll drill down into those and give you some examples of how DNA barcoding is really, really going to be carried out. So in DNA barcoding, the very first thing is that you need to take a sample, let's say, of an organism. It could be a plant, maybe it's an ant that's walking in your driveway, maybe even it's a piece of food, and we'll talk about some examples of what students have done. Then we need to actually do DNA extraction. Now, if we were doing the wet lab, what you would be doing is you would go ahead and extract the DNA, and there's a variety of chemical ways that you could do that. You'd also be using a thing called PCR to amplify that. And if you have some AP Bio background, you'd be able to see that. The next thing is that once you sequence it, and that we'll talk a little bit in detail, then we could go from the sequencing to being able to actually get those ACs, Ts, and Gs. So I'm going to give you some background on that, because the bioinformatics part really does not cover these top things, the organism sample, DNA extracted, barcode. It's really these bottom things, working with the data. And once you have the data, then you can go ahead and actually see the barcode, so to speak. All right, so here is a little bit of history. Let me play you this quick video. And this is Mark Stokel, one of our collaborators, telling you a little bit about the history of barcoding. Then I'll show you some neat examples of what you can do with it, and then we'll actually start an experiment with that. So let me play you the video.
DNA barcoding really started in 2003, and it started with Canadian scientist Paul Hébert at University of Guelph wrote a paper saying that we could use short DNA sequences from a certain region, standardized region of the genome, to identify species. And he said, and we could call these DNA barcodes. And this was really by analogy with, you know, you go through the supermarket and you go to checkout, the cashier doesn't have to read the label they just scan it. And that's much faster than reading labels. So the idea was, could we develop a system for species that would have some of those same features, where we could use a machine to read the DNA, in a sense, match that to a name. And that would help us identify things, which is really difficult to do. There are about 2 million named species of plants and animals, and that's far more than any one person can keep track of. And then in addition, a lot of things have different life stages, their eggs, their larvae, or you might want to know what's in the stomach. What does something eat? When this started in 2003, it was an idea that Paul Hébert proposed, and that led to a workshop, 2 workshops actually at Cold Spring Harbor, one in March and then another one in the following September, to just get other scientists together to say, okay, that's a nice idea, but really, is it going to work? Because at that time there weren't barcodes for many species, and is it going to work? And how, if it's going to work, how are we going to make it work? And from the beginning, it was clear it was a way of taking science, knowledge of experts, and making it available to a much wider number of people. So 2003, it was an idea, and as of today, there are now over 1 million barcode records. That's from 1 million different specimens, each one is which has been identified individually by a specialist. So.
Okay, so let me just adjust something here. Get my lighting back to where it's a little bit better. So in that video, what you were seeing there was just a description, again, of the start of DNA barcoding by Mark Stokel. And where we are with this today is really to the point at which we can use it in the classroom. So let me ask you a question if you're watching, and this one is talking really about the importance of barcoding. How many species can you name? When I ask that question, you know, people might have a number in their head. Do you think you can name 10? Do you think you can name 20? I wouldn't make you write a list. And then I also usually ask the question, how many animals did you name? How many of them are mammals, or did you name any plants or any insects? Usually what I find is people are a little bit biased. We know the familiar species like a cat or a dog or an oak tree, perhaps. But even once we get beyond, you know, cat or dog, right? We might know it's Canis familiaris. But oak tree, did you know it was Cacerus or shark? So we often know maybe a handful of species, and oftentimes we don't know the scientific name for those species. And if we don't know that scientific name, we really haven't unlocked all of the scientific information about it. So it's really, really important that we actually understand this. In fact, one of the first problems that anybody doing barcoding might encounter is that nobody knows how many species there really are. Mark gave some description in the video. We know that mammals, which is what we tend to go and spend time seeing in a zoo perhaps, is one of the smaller groups. There are lots of birds and reptiles, but insects, there's over 1 million. Most of them sort of beetles. And what about flowering plants, the angiosperms? How many flowering plants can you name? There's more than 200,000, 300,000 perhaps species. So really, really understanding what species are out there, especially when many of them are threatened, maybe many of them have not even been discovered yet. That's an important problem that many of us need to be concerned with, because biodiversity really contributes to our understanding of life and the environment. There are lots of great reasons to know that. Also, when we come to describing species, the description itself of what a species is may be a little bit complex. If two things breed, is that a definition for a species? Or what happens like with these ducks that we have here? If they, especially in birds where the male and the female look very, very different, it's hard to tell whether one thing is the same species or not. And then also currently our taxonomic methods to actually look at this different species is quite slow. Traditionally, here is a look at how a taxonomist might describe a plant. This particular plant is one I used to work with called Dioscorea, and it's got lots of Latin names, and so you probably actually have to study quite a bit in order to be able to even break down what does this mean, that staminate inflorescences are auxiliary and they're one or two per axil that are manipulate and fasciculate, and that the panicles, it just goes on, and really, unfortunately, that's a rare bit of knowledge for most people today. So the idea behind DNA barcoding is actually, rather than focusing on some of the more complex ways to describe a plant or an animal, we can actually look strictly at the DNA. And by looking at the DNA and just now comparing ACs, Ts, and Gs, we can actually get a much more simple way to compare and identify species. Okay, so what are we going to use that for in the lab today? Before I get to that lab, what I actually want to point out to you is the DNA Learning Center website. So it's a little bit meta. I'm going to come onto the DNA Learning Center website, but I'm not going to click on the video because that's what's playing right now. We don't want an infinite loop. But if you go to the DNA Learning Center website, let me not do that too quickly. Make sure you caught up with me. Down at DNALC Live, and if you go to resources, what you're going to find are some packets here. I believe these have been posted. So there is a whole packet of almost all the steps that I'm going to go through. And in fact, it's going to be useful to you for parts 1, 2, and 3 of this experience. So be sure that you come there. And there's also some worksheets. So these will be things to watch before and after. There's some additional resources. So make sure you know about that because it's going to introduce this topic and cover it so that you can get this information no matter when and where you're watching. So let me talk about the mosquitoes, which is the goal of today's lab. Here's the problem, and we're going to work strictly with bioinformatics. So we're not extracting any mosquito DNA, but we are going to ask the question, can you tell the difference between larvae, mosquito larvae, so immature mosquitoes that look nearly identical? So quiz yourself. Here are 3 different species. In fact, the genuses are different of mosquitoes. We have Aedes, which is one particular genus, Anopheles, and Culex. And actually, probably if you look closely, you can already tell that there's some difference between them, right? If you look at the Aedes, it's got this nice black and white banding pattern. The shape of the mosquitoes look quite different. If you look at the Culex, look at the actual antennae and the length of the hairs there. Pretty differentiating characteristics, especially if you study them for quite a while. But if you look at the larvae, as it turns out, the larvae look really, really similar. And in fact, actually, for even trained experts, it can be hard to tell the larvae apart. Here is one close-up showing them all. And so you may run into the situation if you're an entomologist, or even if you are wondering what mosquitoes are in your backyard, of what species am I looking at? Now, why would you care? Most of the times when we think of mosquitoes, we may think of them as something that we don't like to have around. But it actually does matter. If we're also talking about epidemiology and disease, these different mosquitoes carry different types of diseases. And so it'd be good to know if any particular area, what species do you have around, and are they transmitting some diseases? And in fact, when we look at climate change, we know that the range of these mosquitoes is different. And so we may expect to see different mosquitoes in different parts of the country, if we're just talking about the US, and that may be changing. And it would be good to know if we see, are there mosquitoes making an appearance that we never saw before? And so should we be testing and should we be looking out for new things, including diseases perhaps that we hadn't seen before? So those are all elements of why you might care about it. And I have some other slides which I didn't put on today, but I'll even show you next time perhaps what some of the things that students have found, not just looking at mosquitoes, but looking at foods and detecting food fraud. So we'll get to that. We'll save that for next time, since they didn't make it into today's slide deck, but the principles will all be the same. Okay, so in order to wonder about these mosquitoes, we need to look at their DNA. And in order to wonder about their DNA, we actually need to understand how we get DNA. So in order to tell you a little bit more about this, it's listed in the references. So if you downloaded the handout, you have it there. Or if you come to the Cold Spring Harbor DNA Learning Center website, you'll come to this description on sequencing. Now, let me remind you and sort of warn you, if you've taken AP Biology and you're pretty far along in the year, a lot of these terms will be familiar. But if you're watching the video and you haven't gotten a little bit of grounding in molecular biology, don't worry about that. In the handouts, there are some references so you can brush up a little bit. But let me give you at least the very minimum on how DNA sequencing works, because that's how we get the data. And it's really, really important because later we're going to talk about some experimental concepts that aren't just part of bioinformatics, but any experiment. We talk about the quality of our data and controls. So that'll be important later. But let me take you through this nice animation on sequencing. Okay, so to sequence a piece of DNA, you need a couple of things. One, you need the piece of DNA that you actually want to read. And normally we get this from a technique called polymerase chain reaction. I'm not describing that here for the sake of time, but just letting you know that this data are here. And so you may want to take a look at that. The next thing is that you actually need the individual components of a piece of DNA, which are the bases, the A's, C's, T's, and G's. We chemically manufacture those. You need an enzyme that's going to do some polymerizing, since this is a polymerase enzyme that's going to build DNA fragments. And we also need a primer, which is a short piece of DNA that gets the whole process started. So let me take you through this. And there's one more quote unquote magic ingredient, we're going to add these other special A's, C's, T's, and G's, these dideoxynucleotides. If you remember, DNA is deoxyribonucleic acid. Well, these are dideoxy, missing 2 oxygens. That's going to be important later. I'll try to at least hint at some of the biochemistry involved. The thing that you could actually focus on for right now is that they all have different colors. So that'll be important in just a moment. Okay, so what happens? We take all this stuff and mix it into a tube, and what do we actually do? Let's jump ahead in the animation. And notice there's a thermometer because we do this reaction at several different specific temperatures. So the very first thing we do is we take our tube and we heat it up quite a bit, around 96 degrees, and that is to separate the DNA into 2 separate strands. So DNA is double-stranded. We need to separate it into single-stranded DNA. What comes next? Well, we cool it down slightly, and we cool it just enough that that primer can bind. Remember, it's a really short piece of DNA, and if you're thinking in biochemistry, when something, or even let's just say in your everyday life, you know that a cup of water that's hot takes a short amount of time to cool down, but a couple gallons of water that was hot, well, that would take a long time to cool down. So by cooling it to this temperature of 50, it's cool enough that that short piece of DNA cools down, it binds, but the really longer pieces of DNA, the other half of this strand, they're not going to bind yet. They're still hot, moving around, wiggling around, so only the primer is going to bind. So we're going to then add just a slight more bit of energy, not enough to break those bonds, but enough for the polymerase that's there to become active. And if you know what a polymerase is, great, but if you don't, there's definitely something you want to look up. The polymerase is an enzyme which actually extends DNA. It builds DNA. So it's going to incorporate those individual bases. And if you notice here, we've got bases. Some of them are brown, which are regular deoxynucleotide bases, but some of them are colored, which are those dideoxynucleotides. We go ahead and let the process run. And what you'll see is that we slowly incorporate those individual bases. But every once in a while, just by random chance, what's going to happen is that we get an A, in this case, which is one of those dideoxy bars. And because it's dideoxy, it's missing an extra oxygen. When the polymerase tries to add the next base, it actually will not succeed. The next base, try to add that, it actually can't connect. And so the polymerase quits, falls off, and you end up with a piece of DNA that is at the end of it labeled with one of these colored nucleotides. Now, what does that get you? Well, let me show you in the next screen here. This process is happening millions and millions of times in a tube, and it's a completely random process. So here you didn't get so lucky. The very first nucleotide you incorporated was one of these dideoxy T's. It stopped, didn't go any further. Here it came a little bit further, got all the way out to a C where it stopped. It happened to be a blue nucleotide dideoxy, and a G and a C and an A, so on and so forth. So what's going to happen is that you're going to end up with millions of strands of DNA that get copied, that get made from this process, all of different lengths, and all ending with one of these colored labels. So a quick question to throw out to you to think about is, does anybody know a process whereby you can sort DNA according to its size? If you've taken your biology, you may be thinking of a process called electrophoresis. So we're going to do that because electrophoresis allows us to separate DNA by its size. Now, if you've done electrophoresis, you may have done it on a flat gel, which is pretty popular today, common, and we still use it for lots of things. But in the case of DNA sequencing, what you're going to use is a gel that's much, much thicker than the regular gels which we make out of agarose. We're going to be using this gel, which is made out of something called polyacrylamide. And so we load it onto a gel, put it into a machine, and there's a number of ways to do it, but this happens to be the one that we're showing. And most of the sequencing, this is called cycle sequencing or Sanger cycle sequencing, works in very similar way. Here's what happens as we go through. We turn on the electricity, as you know, electrophoresis, and those DNA bands are pushed by electricity through the gel. And you can kind of see them, I'm going to zoom in a little bit, as these little bands coming off the screen. And each one of those bands has a color that's read by a laser at the end. And the cool thing about electrophoresis with polyacrylamide is that it's so thick that you can tell pieces of DNA apart, even if they only differ by one nucleotide. And so the result of the laser measuring each of those strands is that you get this readout of DNA sequencing. Now, I will admit the very first time that I looked at this animation and learned about cycle sequencing myself, it was hard to understand. So if you don't quite 100% get it, what this electrophoregram is, we can discuss more in the questions, or we can follow up online. But the long and short of it is that once you organize those millions of random strands into order, if we organize them by their size, then what you're actually doing is figuring out at any one given piece along the DNA strand, you're actually figuring out what the sequence was, what the base is at that particular position. And I would suggest if you get it, great. If it clicks for you in the first time, which it did not for me, wonderful. But if it doesn't, watch it a couple times. And if it still doesn't, maybe that'll be something we answer in questions, or it might also be good for an office hour where I can take out my pen and draw some diagrams with you and work with you to understand it. But just accept that this process is the one that allows us to actually read out the actual A's, C's, T's, and G's. And this is also the point at which most bioinformaticians begin, because they don't usually do that wet lab themselves. I know how to do both, and many people do know how to do both, but you can be a bioinformatician even if you're pretty clumsy in the lab, so that's pretty nice. Okay, so let's go on. We've accepted that there's an experimental way to get us the actual A's, C's, T's, and G's. And in fact, here's just a little bit of the Anopheles gambiae, in this case, mitochondrial genome. Where are we looking at? Oh, this is actually some of the nuclear genome. It's a particular region of a mosquito genome. So there is an experimental apparatus that gives you these A's, C's, T's, and G's. And that bioinformaticians will come to a website like NCBI to download that data and start their work, or perhaps they're working in a lab where other scientists have generated this information and actually use that to begin their experiments. So now what we're going to do, and this is also a great time to take out your packet if you have downloaded it from the DNA Learning Center website, we're going to start a project. We're going to get a little bit into it, and that's why we call this part 1. There's going to be 2 more parts at least where we're going to actually finish the project. So if you have the packet, you might want to get that out to follow along. We're going to go on to DNA Subway. So let me tell you a little bit about that, get you on there, and walk you through it. Again, if you are following along, let's go one last time to the DNA Learning Center homepage. I'm really zoomed in right now. DNA Learning Center homepage, DNALC Live. This is today's broadcast, which is Barcoding Bioinformatics, and under Resources, we've got those packets. The Barcoding Bioinformatics Part 1 student handout is just for students to go ahead and take a look, and some thought questions that you could be able to complete after. The full packet you can download, and it's really where I'm about to take you now, starting on page 4. So let's go to DNA Subway, which is this tool. All right, let me actually log out to show you what it looks like. So this is DNA Subway, and I'm going to zoom in here a bit. And this is an online bioinformatics tool. And what we did in building DNA Subway was take a lot of the bioinformatics software that scientists use to study, whether we're looking at mosquito genomes, whether we are studying and looking at the coronavirus genome, whatever it is, we've put those into DNA Subway to help us analyze this data and make it really easy for you to follow along and analyze this data too. In the future, we're going to have some online courses that go through all of the different bits and pieces of DNA Subway. I'll also remind you down here, there is a manual for DNA Subway. So there's a link that can take you to learning more about Subway. But for right now, what we're going to do is use DNA Subway. And if you want, you can actually register, and that'll take you to get an account. We encourage you to go ahead and get an account, especially if you're going to be using it in the future. But for right now, even, you can go ahead and enter as a guest, and you'd be able to use it as a guest. So I'm going to go ahead, since this is going to be gone over a couple of weeks, and make a project entering as a guest with the password that you're going to get for yourself, well, entering as a guest, your work will not be saved. So that's the more important thing. If you get an account, then when you come back for part 2 and part 3, then you'll continue on from there. So if you want to follow along as a guest, go ahead and do that. And we won't go so far along that it'll take you a long time to recreate what we've done. So I'm going to go ahead and log in. When I log in, I see a whole bunch of projects that I've created in the past. I don't need those right now, but here is the first thing that you'll see. So as you create projects, you'll see them all here. What we're going to do is click on the blue square, which is called Determine Sequence Relationship. So anywhere in DNA Subway, clicking on that blue square is going to help you start. Now, next time, we'll talk a little bit more about the actual piece of DNA that we are looking at in order to be able to determine the relationships between species. But for right now, let's start a project, get as far as we can go, and we'll learn a little bit more in part 2 about some of these genes. We're going to be using a gene called CO1, and it is a gene that's part of your mitochondrial genome. And it's actually a gene that's present in just about all animals. So it's this thing that we all have in common. Not every single animal shares every single gene, but we all do share some genes. And this CO1 is one of those genes that we all share. And in the project packet, which gives you even more background than I'm giving you right now, so if you're curious, you can already be reading through that and preparing for next time, we want to choose the Intro to Barcoding Bioinformatics mosquitoes. So there's another mosquito set down there, but you want to choose this first one, Intro Barcoding Bioinformatics Mosquitoes. And we're going to give this a project title. So I'm just going to say Webinar Part 1 and go with that. When we click Continue, what it's going to do is prepare a workspace within DNA Subway that we can use in order to do the rest of our analysis. The very first part of our webinar today, the lesson that we're doing today, is going to focus on these beginning things where we're concerned about what DNA sequence quality is, how can we determine if we have a good set of experimental data in order to move forward with the experiment. Then the next time we come back, we'll go a little bit deeper into some of the science behind DNA barcoding, and we'll also learn about this tool called BLAST, which allows us to say, well, here's my sequence, and if I want to compare another sequence, has anybody ever found or described something that looks like what I found or described? And then finally, we'll also show you some other tools in part 3 to actually draw some comparisons between different species in using phylogenetics. So the very first thing we want to do is go to this thing called Sequence Viewer. And amazingly, what we have here are some mosquito data, some mosquito DNA. And I can sort of scroll across, and let me give you a little bit more detail. So number one, you'll see that we have two things that are not mosquitoes at all. These are horseflies. And does anybody know, as a question for anybody that might be listening or be a big fan of bugs, where in the tree of life are mosquitoes? Well, they're pretty similar to flies. So there's the order Diptera. And so horsefly is a relative of mosquito, but obviously they're not. And then we have a whole bunch of unknown mosquitoes, and they have these little dashes, 1F1R, 2F2R. We'll talk a little bit about what that means at some point. But for right now, know that you have the DNA sequence. Now, even more amazingly is if you click on any one of these sequences, if you click on the name of it, it will actually show you this readout of the DNA sequence as the electropherogram. Sometimes, unfortunately, there's two names for it, called the chromatogram, chromos from the word color. We can actually see the DNA sequence, and you can scroll around it. So let me give you a little bit more insight as to what we are seeing here. And let me point out something that's really, really interesting, and is going to be a key concept, probably the key concept for today's lab, which is data quality. And I have a slide, and I'll come back to it. But first, let me just at least decode this a little bit. So as I move along, if you want to think about me moving along the x-axis or moving along from left to right, I'm going along the strand of DNA. So here's 10 nucleotides in, 20 nucleotides, 30, so on and so forth. Now, if you look at the top here, you'll notice that there are a whole bunch of N's, which that's not a DNA nucleotide as far as I know. Then you see some T's, and you see some A's and G's and C's and so on. So what is going on with this first half here? Why do we see these things called N's? Well, as it turns out, these things called N means, and if you look in your packet, it explains that the word any, it means it's an unknown nucleotide. In other words, the data here are not very good. This is basically saying that the computer that read this gel when it was being sequenced was not able to determine what the DNA sequence was. Now, if you look down at the bottom when we see these peaks, you can probably by now start to put together that if you have a red peak, you have a T. If you have a blue peak, you have a C, and so on and so forth. So why here, I can clearly see at least a couple T's. Why are we not calling those as T's, or why do we have this jumble? Well, there are some experimental reasons why DNA sequencing, especially at the very beginning of a DNA sequence, and also at the very end of a DNA sequence, are usually lower in quality. We can talk about those, but really it's the word quality that I want you to be concerned with. And in fact, I actually have a slide to help me describe this because it's so important. In fact, actually, let me just go ahead and open what I think was a different set of slides that had a little bit more information. Actually, let's go back this way. And I wanted to really get this across if I find it. If I don't, then I'm just going to tell you what it is because I have quite a number of things here open. All right. So what I wanted to get for you, if this is it. Okay, what I wanted to really get to you is this idea of data quality being a key concept. Now, what we see here, or if we were to look at many, many different ones of these chromatograms, what you end up seeing is that some of them have really, really well-defined peaks and lines, and some of them have ones that are less well-defined. And this is in the packet if you're looking for reference. So down here, we see some low-quality sequence where those peaks and lines are sort of all on top of each other. And in fact, if they are really, really on top of each other, nothing is called. The idea of sequence quality is really, really important because if you think about it, I'm actually going to stop sharing and just talk to you for a moment because it's really exciting to me and it's really, really crucial for people to know about. When you do an experiment, probably by now, you are familiar with the idea of a control, that you have a positive control and maybe you have a negative control. And when I teach about controls, I always teach about controls as controls answer a question for you in your experiment. A positive control, well, what question might that answer? A positive control answers the question, if I were to see an effect in my experiment, what should it look like, right? So for example, if I was doing an experiment on watering my plants and can my plants live without water? If I water plants, or if I have a control plant that gets water every day, well, then if let's say I want to water the other set of plants with vinegar instead of water, well, I have my positive control would be a plant that has regular water, right? And if that grows well, then I'll know that my seeds are good, my soil is good, and now vinegar is the only distinguishing factor. And if I plant if I have some plants and I give them only vinegar instead of water, if they don't grow, but my positive control grows, then I know that that looks great. You could also have a negative control. What would things look like if I don't have an effect? Well, what if I had a plant and I didn't water at all, and then the other ones I watered? Well, the one that got no water, in that case, if I'm asking the question, what do plants without water look like? Will they grow spontaneously? And well, that would be my negative control. But there is one more type of control that you can actually have, and that's called a sensitivity control. A sensitivity control is asking the question, across what ranges can I see an effect? So imagine, I don't have one handy at the moment, imagine you have a thermometer, and your thermometer reads from 0 degrees to 100 degrees Celsius. So 0 degrees is freezing, and 100 degrees is boiling. Great. But what if I were to take that thermometer and use it, let's say making candy? If you've ever made candy, you actually have to be really careful because melted sugar is really hot. And it can be something on the order of in more than 100 degrees Celsius. And the Fahrenheit, I'm remembering this stage, it's around 300 degrees. Well, if you take an ordinary thermometer that only goes from 0 to 100, but what you need to measure is at 300, your thermometer will go all the way up to 100, but it won't go any further, right? So you cannot measure an effect that's higher than what you actually have. So how does that all relate back to our DNA sequence? And how does it all relate back to data quality and controls? Well, here is our DNA sequence. And what it turns out is that when we look at the peaks and we look at the valleys, what we're actually asking, or what the computer is trying to interpret, is given the sensitivity of this computer, giving this the software and the signal that it's getting, is it able to make a good measurement at a particular location? So if you look here, where we have these horizontal bars, which I didn't explain what actually means something here. These horizontal bars are the computer's estimate of the quality of this data and how sensitive it is and whether that data is good. So even though you see these peaks that look pretty clear, what the computer knows is that because of the intensity of the signal, and because these signals aren't very well spaced out, whereas if I go further, these are really, really sharp, well spaced out signals, well, that this data is a little bit suspect. So we don't necessarily have a positive control, negative control situation here, but we do have a sensitivity control situation where we're asking the question, how good is the quality of my data? How sensitive it is? Now, as it turns out, this thin blue line that's here is not drawn randomly. If something is above that thin blue line, it's of good quality, and something is bad below that line, it's of poorer quality. Well, how do we quantify that? Because if we're thinking in science, we should also be thinking about quantifying things. So let me click here. This little question mark will bring you. And in fact, actually, if you take a link, it'll take you to this place called Wikipedia. Now, I happen to have, and you should certainly go to the Wikipedia link, but I happen to have a slide, so I'm going to go ahead and use it. It explains just a little bit more and gives you a little bit of a detail here. When we talk about DNA quality in bioinformatics and as doing an experiment as biologists, we use this thing called a Phred score. So we can label our DNA, whether it was a Phred score of 10 or whether it was a Phred score of 20 or 30 or 40 or so on and so forth. As it turns out, depending on what that score is, we actually say, what's the probability that for any given base with a particular call, let's say we have a Phred score of 10, and the computer has called it as an A, but it said, you know what, this Phred score is a 10, I'm not really certain. Well, that means that there's a 1 in 10 chance that that is actually a mistake. And in fact, that's an accuracy of 90%. Now, in everyday life, when you're talking about things, if I told you that a test is 90% accurate, you might say pretty good. But as people think about statistics, if even right now when people are being tested for coronavirus in that example, that means that if you test 100 people, 10 of them will have the wrong result. So probably want to do better than that. If you could get a test that was 99% accurate, well, now only one person may actually get a result that is not trustworthy. And when you start to go, let me bring up this so that you can see it a little bit closer on my chart here because I wasn't sharing it. There we go. When I bring up this chart, if you were following along on Wikipedia, but I've made it a little bit bigger here. If you have something that's 99%, well, now that is really, really good. But if you think about DNA, and if you think about that many organisms have millions of DNA nucleotides, and in the case of humans, we have billions of nucleotides, you want something that's even better oftentimes than 99% accuracy. So you want to getting out in the range of 99.9 or 99.99. And if you do some math, which will be an exercise that I won't do with you, but I encourage you to take a look at those worksheets, you'll be able to figure out what type of Phred scores would you really, really want to look at for being able to get high quality data that you use in an experiment. So where we should be at right now and what we're going to be doing the next time is a little bit of homework, and it will prompt you and give you some questions to look at. So when we come back next time, we'll continue with the experiment. What you should do is take a look at the data that we have here. And we've got some for all of the mosquitoes. And let me clue you in that when we say a mosquito is mosquito 1F and 1R, those are the same mosquitoes. So we've got 1, 2, 3, and 4 mosquitoes. As it turns out, and as we'll go through in a little bit more depth next time, we sequence the mosquito DNA twice in order to minimize the chance that we have error. So we sequence it twice, and I'll talk a little bit about bidirectional sequencing. And what that takes care of is even if we have some data that is not the highest quality, if we do it at least twice, then we may be able to recover areas where the computer in one particular case did not come to a determinate conclusion, but in another case, perhaps it did. So take a look at those and answer the questions in the worksheet, which are particularly concerned with what do we think is good quality DNA. And when we come back, what we're going to do is in part 2, we will actually pair those DNA sequences, make sure we have the highest quality data, and we actually need that really high quality data in order to do our blast search. So I think that's what's coming up for next time. And I just want to remind you, let me bring up the slides again, if you are following along, to go back and look at those packets and worksheets at the DNA Learning Center website. We'll also, in advance of the part 2, post those packets, especially the worksheets, a new version of it with some pre-lab materials that you may want to take a look at. And because it's the first time we're doing it, we're really interested to hear back from teachers and from educators of all sorts and students on what you would like and if this is helpful and what more we can do. But by the time we get through this particular three-part series, I will really have given you some in-depth look at what DNA barcoding is and prepare you for some of the other things that we have, including being able perhaps to do it at your home or joining some of the DNA barcoding programs that we will be offering and continue to offer at the Learning Center. So with that said, I want to just check to make sure that I don't have any questions. I'm looking off screen here, but I think things have been moderated by my colleagues who've been paying attention to the chat. But you can also, these videos will be posted. So if you have questions that just didn't occur to you now, or you're watching this later, go ahead and post those questions. We'll answer them either directly or we'll come back with those questions next time. So with that said, thank you for watching this very first episode of Barcoding Bioinformatics and this very first experiment in DNA Learning Center Live. And we are going to be producing even more stuff and really look forward to making sure that everybody is still able to continue understanding some of the exciting biology that is at your fingertips with all the resources we offer on our website. So with that, have a great day.